dear students, welcome to Brush Up Zoology, and we are continuing the lecture on immunology basics. So far, in my last lecture, we talked about different type of. We were talking about different type of mechanisms which operate through the innate immunity. As we talked about innate immunity is something which is the inborn immunity, while the second type of immunity we talked about was I gave you an introduction. The second type of immunity is the adaptive immunity. In innate immunity, we were talking about certain barriers, which were the barriers were physical, physiological, and then we also talked about cellular barriers. The cellular barriers includes a number of cells, variety of cells, and certain soluble molecules. So what are the soluble molecules? Soluble molecules are the ones which are secreted by these cells. Amongst the variety of cells which we are talking about are the different type of cells which are involved in these are involved in different type which are involved in the innate immunity or other kind of immunity also. These cells are let's say microphages and macrophages, and third one we talked about NK. So first we'll talk about the microphages, and second type are the macrophages. These two collectively are phagocytes, out of which macrophages, these are, as the name is saying, small cells which phagocytize. The central cell to these macrophages are the leukocytes. Leukocytes are the main WBCs, out of which leukocytes, WBCs, out of which the neutrophils are the main cells that phagocytize. They are generally the micro cells. They are short-lived, about let's say 6 to 7 days their life is, and they are secreted by the stem cells, precursor cells in bone marrow. So here is their origin. They would be secreted in the bone marrow, and they are short-lived, and majority are neutrophils, and they develop from bone marrow. These points we need to remember. And then what they do, in the last lecture also we talked about, as soon as certain injury happens and certain foreign particle, or let's say the pathogen that gets into the tissue, certain tissue, or at the periphery of the tissue, suppose this is the pathogen, these neutrophils are already, macrophages are already streaming in the blood, and they leave the blood capillaries and they surround the pathogen, they engulf the pathogen and they kill the pathogen, and themselves too they die, and therefore they are short-lived. Whereas these another name for these neutrophils, we write them as PMN, polymorphonuclear sites, because their nucleus is like this, something like this. They are granular. I've made the neutrophils. Second type of the cells which phagocytize They are the large cells, which are called as the macrophages. Now, what are macrophages? Macrophages are, let's say, they are also called as mononuclear phagocytes. Mononuclear phagocytes, and they are also derived from mononuclear lineage from the same cells, that is the stem cells in bone marrow. Right? So they also have the common region, but the difference is, one, they are large sized, and second, they are long lived. They live long, and they are of two. Let's say they are, for example, are the monocytes. They are agranular leukocytes, and along with them are the macrophages. So what monocytes do? Monocytes, they are also circulating in the blood. So monocytes, suppose this is the monocyte, they leave the blood. They are already in the blood. They leave the blood and they get into the tissues, where they get converted into macrophages. And macrophages are the cells which are ready to, let's say, attack the pathogens. They engulf, what they do, the macrophages, they engulf the foreign particle. Any kind of foreign particle that can be a pathogen too. And the strategy is such of our physiological system. Macrophages are present in the where they are actually required. So, for example, within the tissues, Kupffer cells present, they line the sinusoids. Similarly, another example is synovial A cells. A cells, these line the synovial cavity. So that was about the macrophages and how they behave. Macrophages, they engulf large particles and then they destroy them. The strategy is same, and the main strategy is where they are located. They would be located, some such as synovial cavity, lining the synovial cavity. Synovial A cells are located, where similarly Kupffer cells, they would be located in around the sinusoids within which the blood flows, and there are more chances that they encounter the pathogens at those sites. So macrophages, they are mononuclear, they are large, and mostly they are irregular. They are phagocytic cells. And then we have another different kind of cells which are called as B cells and T cells. Although they do not directly play a role in the innate immunity, but generally these cells, innate and adaptive immunity, basically they work together. So B cells and T cells, they also have to play a role. What they do, the B cells are the lymphocytes. They are basically the lymphocytes. B lymphocytes, T lymphocytes. B lymphocytes, B T cells, these are lymphocytes, they recognize the pathogen. They, both B cells as well as T cells, their role is to they recognize the pathogen, and so these initiate, let's say, the adaptive immunity. All lymphocytes, they are derived again from the same cells, bone marrow, stem cells. Within bone marrow, they are all lymphocytes, they are derived, but their development is different. B lymphocytes, B cells, they mature in bone marrow in adults, while T lymphocytes, they mature in thymus gland. So, how they behave now? The B lymphocytes, B cells, they express antibody. They have a surface receptor. B cells, they have a surface receptor which is specific for a particular antigen. For an antigen, so that is the specific receptor is basically the antibody. And when B cell happens to come in contact with the antigen, they multiply. And when they multiply, they form a clone of cells which are called as plasma cells. And plasma cells, these plasma cells, they produce the large amount of antibody, right? So this antibody is very similar to the one which is present on the surface of the, which is surface receptor over the B cell. But these antibodies, they, when the plasma cells multiply and they start producing this, the kind of machinery, and these start floating in the blood. They're in secreted form, which are basically large glycoproteins which are found in blood, and this is, you can say, this is the soluble version of this antibody. Originally receptor molecule, whereas